students. Now we can discuss about Sscp. This is a PCR based technique, and here this is a powerful tool to detect single nucleotide polymorphisms or point mutations those are present within the genomes. So we can go what is the basic methodology behind this Sscp. In this picture, here you can see two amplified products. One is this one, and another is this one. Both of them have been obtained from two different samples, but the difference is that here you can see here in this amplified product at this point there is a point mutation that indicates as a block. Then after the PCR reactions, what you have to do is that you have to bring the temperature up. At that temperature, your double-stranded DNA of the amplified products will be denatured into single-stranded DNA. Here you can see that. Here also, the exactly similar things will happen. Then after that, you have to bring the temperature down rapidly. So at that temperature, each of these single-stranded DNA will be folded into like this. So this single-stranded DNA will be folded into a conformation like this, and this single-stranded DNA will be folded like this, and here also this a single-stranded DNA will be folded, but these folds or these conformations may be differ from that of the wild type because it having a single nucleotide difference with that of the wild type. So this single stranded DNA will be folded, but this conformation is quite different from that of the wild type. So here also you can expect a differential type folding. After that, you have to load these products into different lanes of a gel and need to carry out this electrophoresis. After the electrophoresis process, you will get two bands in the gel for sample 1, and for the sample 2 you can expect two results. This particular band may be the resultant of this conformation, and this strand and this band may be the resultant of this particular conformation. And in the mutant type, you will get two bands. The positions of these bands may be different from that of the first line. That indicates the polymorphisms of the two different samples. Now we can move to a modification of the Sscp, that is fluorescence based PCR Sscp, that is FSSCP. Here what you have to do is that the methodology behind this process is exactly similar to that of Sscp, but the difference is that you have to amplify this particular region of the genome, and but the primer sets you had been used here in this reaction is linked with a fluorescent molecule. Suppose this is the forward one, this is the reverse one, and this forward is linked with a blue fluorescent labeled molecule, and the reverse one that is linked with a green one that is indicated here, blue in the forward that indicates here. So you will get the amplified product. In that product, on both ends, you will have fluorescent molecule. This is also an amplified product which you have got from a different sample. This is the amplified product from sample 1, This may be an amplified product from the sample 2, and the sample 1 and sample 2 having only one difference between these two, that is a point mutation at a particular region. Then after the PCR process is over, you have to keep the temperature up. Then at that temperature, your product's double stranded DNA will get denatured into single stranded DNA. You can see this is single stranded DNA and this is another one. And this is single stranded DNA and this is the complementary strand of that double stranded DNA. Then after this denaturation process, you have to keep the temperature down immediately. And at that temperature, each single stranded DNA start to fold into their own way. Okay, so this may be folded into this particular conformation, and this will folded like this conformation, and this single stranded DNA will be folded like this, and this one will be folded into this conformation. All of these conformations having fluorescence in it. After this, you have to carry out the capillary gel electrophoresis, and in this capillary gel electrophoresis pattern, you have two bands. One shows a blue fluorescence over here and another shows a green fluorescence over here. Blue fluorescence is that of this particular conformation, and the green one indicates the presence of this particular conformation. And with this in the capillary gel electrophoresis, you may get different pattern. Sometimes the blue may be over here, but the green may be over here. So you can expect polymorphisms